talking of lipid peroxidation, it is basically causing, it does a lot of things, and there are there are three steps to it. We'll see those steps in details. But one of the important things is it causes cellular damage. वो cellular damage करता है, and that is bad. There are different ways of cellular damage, cell death, that is there. And this is a timeline of the different mechanisms that were identified of how that cell damage is happening. So you have way back in 1965 when programmed cell death mediated by molecular machines were identified. Then you had autophagy, apoptosis, and in all of these, likewise you see with, till recently you have in 2023 disulf disulfidoptosis, in which a new forms of cell death is there because it is triggered by disulfide stress. You have copper stress, ferroptosis, entosis, parthenitos. So many new names are there. At the heart of all of this, you have oxidative stress, lipid peroxidation, and death. You may have heard of apoptosis, ferroptosis, pyroptosis, all those things. There's a lot of research which has happened. There are newer and newer things coming up. A lot of new terminologies are there, right? So that is why it is important for us to know about lipid peroxidation and the kind of damage it has. So we'll move ahead with what lipid peroxidation is. Basically, it is damage to the membranes of the lipids form our cell membranes, and the reactive oxygen species are formed. It basically targets the cell membranes and or membranes of the organelles also. It is basically damaging when it's damaged. The cell membrane, there is oxidative stress, oxidative damage, free radicals are formed, and obviously the body has its own mechanisms of countering these kind of oxidative stress or radical induced damage. But whenever there's an imbalance due to lipid peroxidation, the damage is cellular, the tissue damage increases, and then it builds up. So one of the causes of, in one of the theories with aging, with cardiovascular risk in relation to the biology of cancer, lipid peroxidation plays a role, right? So we should know about lipid peroxidation and try to prevent it. So how does lipid peroxidation happen? Basically, it is you have a lot of unsaturated fatty acids, you have reactive oxygen species which are targeting those double bonds in those unsaturated fatty acids, and then they interact with other things. It is not just restricted to the lipids, it then interacts with the DNA, it interacts with the protein, and it causes damage to those structures also. So a lot of research has happened in the last 4-5 decades that you could see from the timeline that we saw in the beginning. So you have heard of malondialdehyde as a marker of oxidative stress and damage. So for a long time, it is one of the stable markers of lipid peroxidation, and it can be measured. So way back, I think since the last 15-20 years, a lot of research has happened with malondialdehyde as a marker of oxidative stress. So, but then it is not the only marker. There are newer and newer markers which are coming up for oxidative stress, and focus can be given to those markers also. So some of the examples are, it's from malondialdehyde, we realize that there is free malondialdehyde, then there is protein-bound malondialdehyde, total hydroperoxides, which all are representing different kinds of lipid peroxidation and protein lipid peroxide interactions. So there are different methods by which you can measure them. We will not discuss them right now, but then if you are interested, you can have a look at those also. So lipid peroxidation can be considered as a specific process of lipid metabolism. There is excessive lipid peroxidation, which contributes to plasma membrane damage. It plays an important role in mediating. So there is cell signaling, intercellular signaling, lipid peroxide, the metabolites which are formed during lipid peroxidation. Also for the cell in intercellular signaling is there. And how do you stop it? When you think of lipid peroxidation causing so much damage, you have antioxidants, the common vitamins that we use, vitamin A, E, and C, they are all antioxidants. So there are studies where they have seen that exogenous supplementation of vitamin E or tocopherol helps in reducing the cellular damage caused by lipid peroxidation. This here is basically the three steps of lipid peroxidation, which begins with initiation, propagation, and termination. So there is a radical, the radical is there, oxygen radical is there. There's an electron which binds to it. There is superoxide formation, peroxide formation. There is Fenton reaction. So this is part of the Fenton reaction. You have a metal, mostly iron, which is commonly available, which is connected directly with the Haber-Weiss reaction also. And finally, you have the catalase and other enzymes which prevents the lipid peroxidation, and you have the free radicals being converted into a water molecule. The thing is, it can be enzymatic and non-enzymatic. There is initiation, propagation, and termination. Initiation phase, there's a lipid radical denoted here as L with a dot. There is oxidative stress. There's a redox reaction which is there, and there is iron. Iron, in mostly in the ferric form, is involved in lipid peroxidation. And in the propagation steps, what happens is the molecular oxygen gets transferred to the carbon in the mostly the carbons which are associated with the double bonds, and this generates a peroxy radical. Then that is subsequently acted upon by a hydrogen atom or H plus ion, and then a hydroxyl group is formed. Finally, there's one more H which is added. Then you get the water molecule, which is released as a water molecule. So molecular oxygen can be electrochemically. So this process eventually what it does is reduces oxygen into water with the use of 4 different electrons, right? Intermediates are the peroxides, and to counter all this, you have the catalase group of enzymes, and there are 2-3 other groups of enzymes. You have polyphenols, flavonoids, and so there are different mechanisms by which you can prevent it. Now coming to the third part of this picture over here, C, you will see that phospholipid hydroperoxides are formed, and they generate a lot of free radicals by the Fenton reaction. So whenever there's Fenton reaction, you have to remember there is iron. Iron is involved, right? The phospholipid free radicals formed by this process, yeah, can further react with other polyunsaturated fatty acids to increase the chain for the propagation phase. So these are all different pathways and different steps in the lipid peroxidation. 2 important reactions we have to remember here are the Haber-Weiss reaction, where you can see here hydrogen peroxide being converted to oxygen and hydroxyl radical. And in conjunction, you have the Fenton reaction, obviously taking part here, and as well as with the phospholipids. So if you look at the reactions, there is in the initiation step, there are generation of free radicals which take place. They identify the bonds which can be saturated. It elongates, so multiple free radicals are generated in the propagation phase, with a hydrogen being added in the propagation phase. And finally, you realize it is terminated with the release of water or oxygen, where you have a hydroxyl group which is added. So this is the core of lipid peroxidation. Now, this is basically the thing that we saw. Termination concludes when 2 radicals combine to form a stable non-radical product, halting the entire cascade of initiation and propagation of these steps. This is the Fenton and Haber-Weiss reaction, which are basically complementary. You have they are always happening hand in hand. You have the free radicals, the oxygen, the free radical joining a hydrogen ion, forming a OH group, and another H plus ion, forming a hydrogen peroxide group. In Fenton reaction, it reduces it, forming a hydroxyl group and water. Whereas when you're looking at the Haber-Weiss reaction, again, so if you see here, M is metal, N plus 1, so M is transitioning from the ferric to the ferrous form. So 3 plus to 2 plus, and you're converting the free radical to oxygen and water. So when these couple kind of reactions happen, at the heart of all lipid peroxides, peroxidation reactions, you have to remember Fenton and Haber-Weiss reaction. Okay, the thing is, these are important because it causes damage to our membranes, not just the cell membranes, also the organelles, the membranes and the organelles, and it is causing a cascade of signaling and cell death, various kinds of apoptosis that are associated with lipid peroxidation, and also you have seen that there's a flow. I mean, there's a table later on which I'll show you where you can see that there are different kinds of cellular damage mechanisms by which cell damage is done, where lipid peroxidation is at the heart of all the problems. Lipid peroxidation also gives rise to secondary products which causes further damage. So the thing is, you have hydroperoxides, epoxides, which participate in the propagation, and then you have other free radicals which then are not restricted only to lipids, but then interact with proteins, DNA, and also play a part in cell signaling functions. So these are toxic and they are not restricted only to the location where the damage is done. They have extensive signaling mechanisms that are there, and that also causes inflammation and further rise to more and more free radical generation. And there's a cascade which needs to be stopped either with the help of the antioxidants that are present. So we'll also have a look at the antioxidants that can help in stopping these kind of so that it doesn't just go away as a runaway train, and we can stop all these reactions. So the most regarded secondary product is something known as hydroxynonenal 4-HNE, formed as a late product of oxidation of linoleic acid. Uh, and then it is basically kind of an aldehyde with a very high reactivity. And like I said, it reacts not only with the lipids, but also with amino acid residues and DNA bases. And therefore, it has a role in signaling. It is not just 4-HNE, 4-hydroxy nonenal that is there, but there are other similar molecules which are formed, which are again different kinds of short chain aldehydes and ketones. And they again act on different amino acid and uh, DNA groups that are there. So the most important ones that has been studied in the last 3 or 4 decades are oxynonenal and malondialdehyde. So these are more stable and can be studied. So they have been studied by different mechanisms. You can do it by HPLC or by chemical derivatization with thiobarbituric acid. So they have a assay known as the T-bars assay. Formation of electrophiles as late products of lipid peroxidation. We have already seen 4-HNE, 4-hydroxynonenal. So they have this reaction. This is a lot of chemistry. You need not know the details of it. Only thing is, origin is linoleic acid, and then you have a 4-HNE which is formed. This is the structure of it. Then it can also be derived from Fenton reaction of different dihydroperoxides. They eventually are forming malondialdehyde and other carbonyl compounds. So MDA from isoprostane. So the arachidonic acid eventually gives rise to the malondialdehyde, and then different compounds like hexanal, decanal, nonenal. All these are active side chains that are there which participate in different downstream reactions. We need not know the details of all that, but even if you know that, okay, you have something like uh, hexanal or decanal, different carbonyl compounds, MDA, bridge peroxides, Fenton reaction, all these things are important derivatives of or secondary products of lipid peroxidation that happen. Isoprostanes, we have already seen, they are critical. They also play a role in anti-inflammatory activities, but some of them also have a pro-inflammatory function. So among them, you have the 8-API PGF2 alpha, also called as 8-isoprostane. It is an epimer in the C8 form. You don't need to remember all these things. It is an established marker of oxidative stress. So like malondialdehyde is there, you have isoprostanes also, which can be used as markers for oxidative stress. So this is basically an image showing how these lipid peroxides, along with, I mean, react with different amino acids at different sites, like the 4-hydroxy nonenal compound. They're reacting with the amino group. There's always a thio group which is there, and it gives rise to different kinds of adducts. You have a cysteine hydroxy nonenal adduct and a lysine hydroxy nonenal adduct. So there are protein cross-linking which are happening. This is the mechanism or the molecular changes, structural changes, where by which it causes different signaling along with interaction with the DNA. The consequences, obviously, structurally is going to damage the membrane, so it will have an impact on the integrity and function of the organelles. It plays a role in cell signaling. It is associated, like we discussed in the beginning, with aging, cardiovascular disease, cancer, apoptosis, neurological disease, and so on. So there are multiple actual clinical implications of lipid peroxidation that happen. Now coming to antioxidants, free radicals and oxidative stress. Free radicals are, as we know, unstable atoms. You cannot have free radicals stay in that form for long. And the environment plays a role. So whatever environment it is in decides, I mean, determines how long a free radical will be there, because it is always trying to complete a reaction and get stabilized. So to achieve stability, they aggressively seek out and steal electrons from other molecules, damaging vital components. They are not bothered. जहाँ से भी मिले उनको मिलना चाहिए. So causes of free radicals, it can be mainly from normal metabolic processes, body converting to like oxidation that happens at the cellular level. It may also be due to exposure to radiation, pollution, carcinogens, chemicals, tobacco. So all these things are there. When free radicals accumulate faster than our body can get rid of them, obviously that adds to the oxidative damage that happens, and that is why we need antioxidants. When you talk of antioxidants, we have already seen there are vitamins. There are other molecules structurally, I mean, which are there in nature. Things like polyphenols are one of the most commonly available antioxidants in nature. When you talk of sources, you will get these polyphenols in fruits and vegetables, which are easily available. That is why it is recommended to have fruits in your daily diet, right? So antioxidants are basically, again, like we said, group of compounds. They are heterogeneous. How do they interfere with lipid peroxidation? Is basically by preventing I mean the peroxides from developing or breaking a chain when in the propagation, initiation phase, propagation phase, and the termination phase. Recently, there has been some research with the termination enhancing antioxidants. They are belonging to the terpene groups, which, I mean, it's a good to know kind of fact. Otherwise, most of the polyphenols and other antioxidants work by stopping the initiation and propagation of the lipid peroxidation. Terminating the termination, stopping of termination, I mean termination is acted upon only by these terpenoids and essential oils. So it's a novel mechanism that they have found out recently. The first part, the preventive antioxidants, these are again different kinds of molecules. They may have complex enzyme systems. They prevent the initiation of the lipid peroxidation by preventing the formation of initial alkyl radical, which gives rise to the next step, the propagation cycle. What are these molecules? These are like enzymes like catalase. Then you have the thiol peroxidases, which are there. So you need a SH group is closely associated, especially when you talk of glutathione. So SH groups plays a very important role as an antioxidant. Then you have other enzymes like superoxide, dismutase, SODs, glutathione reductase, and different metal chelators. Commonly available metal chelators in our body are transferrin, ceruloplasmin. So they also have an antioxidant function. So you realize, if you think about it, transferrin and ceruloplasmin are often increased when there is acute inflammation as an acute phase reactant. These guys are also increased as an acute phase reactant because biologically it is trying to get rid of those oxidative, the the oxidative stress, reduce the oxidative stress, and they are basically antioxidants. Then the chain breaking ones, also known as radical trapping antioxidant, because it traps the radicals, the free radicals. These are the biggest class of small molecule antioxidants, and here you have the phenols. Phenol, the vitamin E is a tocopherol which has a phenolic group, and it is commonly used. I mean, our body uses tocopherol kind of derivatives for reducing the oxidative stress. So the radical trapping antioxidants act by trapping alkyl peroxyl radicals, thereby competing with the chain propagation and preventing the chain from getting longer and longer and developing newer and newer free radicals, right? So the reaction of the radical trapping antioxidants with the free radicals must be quicker. So it's a competitive kind of situation, like we discussed in a few slides before, where if the oxidative stress is so much and so quick that our body cannot stop it, then the damage is more. Whereas if it is slow, our body has mechanisms of reducing or preventing damage by interfering at these levels. So this is the chain breaking antioxidants that are there. Then there are three different groups also which we should know of. They are polyphenols and flavonoids, sulfonic acid and selenic acids and their derivatives, and aromatic amines and diaryl amines as these radical. So when you talk of polyphenols and flavonoids, you have hydroquinone and catechol as examples. Both these molecules are able to trap the oxygen radicals, the free radicals of oxygen, in a stepwise transfer, finally forming hydroxyl group, and water is released. So phenols often are associated with um, carbon ring kind of group, and these are the commonly used antioxidants by our body. Then you have sulfonic acid, selenic acid derivatives, which are commonly found in a plant which belong to the allium group, and they are also considered a rich source of antioxidants. So the second group is the thio group is there. So they are known as thiosulfonates, like allicin, petiversin. These are available molecules in nature which are potent antioxidants available in various fruits and stuff. Finally, you have the aromatic amine group, which their active side chains, they form the diaryl amines, and they are the second most active antioxidants after the phenols. So there are certain images that you would see here, where you can see this group. You have the catechol group, and the catechol group is reacting with the free radicals. And finally, you have the hydroxyl group and release of uh, a less radical form of the free radicals, right? So this is one of the mechanisms. Second is the tocopherol group, where you have the reduced forms of the tocopherol. Polyphenols and tocopherol is there, and you have in the C compartment you have the sulfonic acid derivatives. Allium cepa is basically onion. You will get something like onion, and it is there in onion which reduces all these kind of oxidative stress. Then you have the aromatic amino acid side chains which help in breaking the chain in the propagation phase and chain termination. So these are different mechanisms by which it is reacting. And if you even understand at least the names and how they're acting at different levels and the compounds which are important as antioxidants, that would help us, I mean, when we're writing an answer in the question. This picture here is to highlight different pathways that are activated. So you can see that there is ferroprocess, reactive oxygen species, the PUFAs are there, lipid peroxidation is there, membrane destruction, ferroprocess. Similarly, when it is involving a cell organelle in the mitochondria, releasing various intracellular, uh, signaling molecules, you have something known as the apoptosome. Caspases are released, substrates are cleaved, apoptosis is there. Inflammatory molecules are also activated via lipid peroxidation. You have vitamin E, which is preventing. You have other antioxidants which prevent the elongation of the chain, but necroptosis, pyroptosis. So this is a polyadenosyl PARP inhibitors are there, which are used as anti-cancer medicine. So there is interaction of reactive oxygen species, lipid leading to lipid peroxidation and DNA damage. So these are different mechanisms by which lipid peroxidation causes damage in different compartments of the cell, different compartments, different kinds of tissues, and their signaling pathways, and how it is important or how it can be prevented by understanding at what level it can be done. Finally, we talk of nitric oxide. Nitric oxide, as we know, is one of the good characters, causes vasodilation, it increases the blood flow, increases oxygen delivery and everything. Broadly, there are two, three types. You have endothelial nitric oxide synthetase and you have an inducible nitric oxide synthetase. These are enzymes which act on arginine and finally it forms nitric oxide is generated. It is one of the commonest gases and it plays a very critical role in G protein coupled receptor mediated signaling. And you can see here that it is also regulated by other neurohormones like acetylcholine, bradykinin, histamine, right? So whenever we talk of nitric oxide, the first thing that comes to our mind is vasodilation. And you have to understand that it is a cascade. So endothelial nitric oxide synthetase can be activated by in calcium dependent or independent manner. So on the one hand, you have the stimulants or agonists like acetylcholine, bradykinin, histamine, which act on specific receptors on the endothelial membrane to increase intracellular calcium concentration. This calcium then binds with calmodulin, leads to activation of calmodulin binding domain of the nitric oxide synthesis to produce nitric oxide. So this is how nitric oxide gets generated. So if you think about it, there is vasodilation, histamine causes vasodilation, bradykinin causes vasodilation, and this is how it does it at the cellular level. On the other hand, phosphorylation of the endothelial nitric oxide synthase independently of the calcium levels is also another mechanism of activation of the enzyme. So there are different activation sites in the enzyme structure, and depending on the pull or push of the pressure or shear stress on the endothelial cells, these things get activated. Now, there is a lot of actin and other molecules which are involved in these kind of signaling pathways, but it is independent of the increase in intracellular calcium. The production of nitric oxide from L-arginine by nitric oxide synthetase requires presence of different cofactors like tetrahydrobiopterin, flavin, FAD, FMN, calmodulin, iron heme. So these are important cofactors. The three nitric oxide synthetic isoforms, they are synthesized as monomers and they need to form dimers in order to bind to this tetrahydrobiopterin. There is another form, a neuronal specific nitric oxide synthetase, which produces nitric oxide both in central CNS and peripheral nervous system. And here the purpose is not vasodilation, but it acts as a uh, neural communication between the nerves. So nitric oxide produced by the nitric oxide synthetase is implicated in regulation of neuronal excitability and firing. And there is something known as neuronal plasticity or synaptic plasticity, which plays a very critical role in our learning and memory. So if you think of nitric oxide, it has multiple functions, the two major ones being vasodilation and associated with dementia. So these kind of things are important in the management. And when you think of it in the context, you have lipid peroxidation creating damage by activating multiple pathways. The body has its safety mechanisms by antioxidants and different kinds of which some of them are synthesized in endogenously in the body, like ceruloplasmin, transferrin, and so on, and others need to be procured from outside. Uh, Polyphenols, flavonoids, and then you have the thio group, which are there, metal ion chelators. These all form our defense system against the free radical induced lipid peroxidation that happens. So with this, I will end